Okay, so today we are going to talk about Nirenberg and Mattei versus Nirenberg and Leader in their genetic codon experiments. And so we'll start with Nirenberg and Mattei. So they basically wanted to determine what three-letter combination would code for what specific amino acid. So they had a combination of poly-U, so a string of poly-U's. And then they had cell-free extracts, so they had a cell and they took everything in it and they put that into a tube along with the poly-U. And then they took each of the 20 amino acids and they would put those into the tubes. So they repeated this 20 times in order to cover each of the 20 amino acids. And the only radioactive amino acid that they had present was the phenylalanine. Sorry if that's wrong, but that's the only one that was radioactive. So then when they repeated this experiment again, they had poly C, and then they did poly A, and then they did poly G. And poly C, proline was the only one that was radioactive, lysine was the only one for poly A, and nothing worked for poly G. So they were only able to find 3 of the 64 codons. And these tubes right here, or this image right here, just kind of an example of like what the tubes could have looked like for each of the proteins. It's nothing in particular, just wanted to add a little visual. And then Nirenberg worked with Leader, and he was able to kind of come up with the 61 remaining codons that we have. But they used tRNA in their experiment. So they had a combination of short mRNA strands with different combinations, codon combinations on there. And then we had ribosomes, and then we had tRNA with amino acid attached. So this image on the side, this is just a representation of the tRNA attached with the amino acid, nothing else in particular, just kind of wanted to show that visual. So they repeated this experiment several times with different radioactive amino acids. But once they had this combination made with all of these 3 different kind of aspects of it, they waited for binding to occur between the tRNA, the ribosome, and the codon. So if there was binding, it would attach to a filter paper, and if there was no binding, it would pass through that filter paper and nothing kind of would attach to it. But if there was an attachment to it, that just meant that there was a radioactive protein present, and then whatever codon was used would mean that that specific amino acid kind of corresponded with that specific codon sequence. So that's how they were able to determine the 61 remaining genetic codons. And that's kind of a short summary of both of those. So thank you.